He loves my bird had like one lab drew a crab.
What are we talking about? One lab you drew a crab.
Oh, for physiology.
An interesting lab. So when you walk in the door, like when you walk in from outside this way, there's a little holder bolted to the side of the building that I have now. So you give that to him, go to the main office, and Miss Christie in the main office has some. Yep.
You're gonna be good.
Okay.
The bookstore, I got a book bundle and they sent me the wrong one. And yesterday I tried to go exchange it and no email got sent. Try it again today.
Send me an email.
Okay.
I will scan like the first chapter for you and send it to you so that you're not behind.
I did this morning a trial access for 21 days. So it let me do the first chapter because I looked at that. I just don't know how long, how many chapters it's going to give me because it did say it had a limited amount.
Yeah. Well, so one chapter will give us a whole week.
Okay.
So if it runs out, if it doesn't give you whatever it doesn't give you, email me right away and say, Dr. This is what's going on.
Yeah.
And I will fix it.
Okay, because I said to go tomorrow and see if they can do it.
Do what they tell you to do, but whatever happens, you tell me what's going on.
Okay.
And I'll help you.
All right, thank you.
Yeah. Well, one instance of iClicker can run at a time. Okay. We got another 6 minutes. If you know about iClicker, how many of y'all have used iClicker before? Some? So if you know about iClicker, it's just attendance. You don't have to buy it for this class. You don't have to buy it. It is just attendance. So I know it's up there. I just don't want you to think, oh my God, I got to go buy it now. You don't have to buy it. You have to download the app or use the handheld iClicker. So you can either do it on your phone or you can use the handheld, but you do not need to buy it. Cool? All right. So y'all keep talking. We still got 5 minutes. There's no code. I think you have to, if you're going to do that, if you go to Moodle and then register your remote with the class, then it should. And I'll tell you the backup for if you can't today. So don't freak out. There's a backup. There's always a backup. So that's for whoever's on Zoom. There. Now you can kind of see me. Yes. Oh. On a hand there.
I think you have. You don't.
Yeah, Reach is easy. It's free on your phone. Like you're never going to forget your phone. Even if you have a handheld iClicker, you don't have to use the handheld iClicker. You can create an account on iCloud and use the app for free.
So it's just strictly for attendance?
It's strictly for attendance. That's it. That's all we're using it for.
He's got black hair. They're relevant 28, right?
join the, like, is there a certain section we need to join?
There's only one section of 435,
Okay.
so it should be fine. You might see my 109 class in there, but.
I think I got it, thank you.
Good start.
Yes, ma'am. Let's see what we're set at. Does not say on the base. Try AA. Try AA. There we go.
Oh, I see. Thank you.
My watch and my phone are different. My watch says 10:44. My phone says 10:42. 10:42? Okay, so my watch is 2 minutes fast. It's good to know.
We like 3 hours. 3 hours.
Yeah.
Yeah, the app is, I find the app easier.
Yeah, I don't know how to do the clicker, so I just got the app.
I mean, the app is just, you're always going to be carrying your phone. And all I'm using it for is attendance. So to me, that's easier. Like if you have your phone, your iPad, your computer, any device, you can check in on attendance. I think it's AA.
It is.
It is? Okay, AA. If it's not working on the handheld iClicker, you can use your computer, you can use your phone, you can use the Reef app on your phone, or you can go to iCloud.com and you can just, it's just attendance. But, so it's free, you don't have to buy the app, it's just attendance. I can't hear you. Yep, the Reef app. Yours says you are.
Refresh it. Yes. Refresh it. Exit out of that screen and come back to that screen. So you go to the
Okay. Looks like it's about time for us to start. So welcome to Biochemistry 1, Chem 435. Everybody in the right place? All right, so I always start out class with a joke. In case you don't know, I see so many familiar faces. It's so much fun to see familiar faces. So in case you don't know, I have a son. He is 9. He is in the 4th grade, and we are on the joke telling kick. So all of my jokes are of the quality of 4th grade. Putting it out there now. All right, so what kind of eggs do evil hens lay? Deviled eggs. That's good, right? That is the quality you should expect for the rest of the semester, okay? I'm not promising anything more. All right, so first thing that I want everybody to do is to try to sign in with the iClicker Reef app. Now, this is the free version. You do not need to buy it. I'm only going to use it for attendance. So anybody not used iClicker before ever in the past? Oh, good. Yay. So the easiest thing I have found is to use your phone and to use the app. You can use the handheld iClicker, but if you use the phone, you're never going to forget your phone. It's easy. You're going to log in. You're going to know. It's just easy. So it doesn't matter to me. If it does not work for you, if you do not see your name here as present, and it does not show up as present on your screen, before you walk out today, you need to sign your name. Now, if you see your name up here on the screen, you see present, you don't have to do anything, okay? And I'll try to remember everybody before I dismiss everybody. But if it doesn't work by the end of the period, this is where you sign up that you are present. Okay? Yes, ma'am.
Can you scroll down?
Sure. Do a slow scroll so everybody can see their names, maybe. It's most likely, I think the box is AA. Why will it not go down? Okay. Everybody good? All right, and I'm happy to check at the end of the period for anybody. I'm just going to go ahead and hide it for now, just so we can see. Now, I have a Zoom going for anyone who is sick, anyone who is in quarantine, or has student access accommodations. But it's kind of long all the way in the back, so I know it's kind of hard to see the screen sometimes. So I am okay if you are in the classroom and you want to join the Zoom so that you have that screen right in front of you. Okay, so if you want to bring your computer, you want to bring your iPad, you want to join the Zoom so that the board is basically right in front of you, I'm okay with that. Just please remember that you should be muted. Let me minimize this. You should mute your microphone. You should mute your input. Otherwise, we're going to get some crazy feedback and it's going to sound horrible in class. And I try to leave the chat up, which I say that and I didn't yet. I tried to leave the chat kind of in the corner so that y'all can use the chat function also to ask questions, because I know sometimes it's hard to hear with the mask and everything going on. So, but always just stop me if you have a question. Okay, so who am I? I am Dr. Sarah Bergeron. I go by Dr. B. So that you know a little bit about me, I have a Bachelor of Science in Microbiology with a minor in Chemistry from UL Lafayette. I then went and got my PhD in Molecular and Cellular Biology, but it was from a biochemistry lab, so all the research that I did was all biochemistry at the University of Iowa. I tell you, Iowa's cold. That was a shock to my system. I am from here. I was actually born in Houma. So Iowa was a shock. And then that wasn't enough. So I went back to school again and I got a Master's of Art in Teaching in Secondary Science Education from right here at Nicholls State. So I've kind of been everywhere, even here. So this is me telling you that I am going to support you. I have your back in a good way. I have your back. I try to be really quick on email responses. Whatever you need, please, please, please let me know. I'm going to show you a bunch of different ways to get into contact with me. But if you don't tell me you're struggling, I don't know. Look how many people are in the room, right? If you don't come and ask for help, I don't know that you need help. If you don't speak up in class, oh wait, Dr. B, we didn't get that. Please say it, all right? So please, please communicate with me as much as possible, and I'm gonna try and help you out as much as possible. Ask anybody who's had me in the past, okay? But you have to tell me you need help. So that is Mr. Charles, my son, up there in the corner. who is into jokes and Legos currently. And then this summer, y'all, I'm so excited. This summer we got a dog. So she is currently 4 months old in that picture. So that was like last weekend. She's a Great Dane.
This is the smallest.
This is the small. She's 40 pounds at 4 months.
Oh, goodness.
So she sometimes comes to campus with me, so you might see her every now and then. But I got to teach her not to bark. Oh, and we're not doing that. That's only for my 109 class. Okay. So that is our little intro. Now, what I want to do is I kind of want to walk through a few things on our Moodle site, just so that you know where everything is located, okay? The very top when you log in, here's all my contact info. This is my office. I'm right down the hall. I'm only doing virtual appointments this semester because of COVID, right? This is why we're all wearing masks. My office is small. We're not going to be able to stay very far apart. So I'm doing Zoom appointments. And if you tell me ahead of time, I usually know that there's an empty room somewhere. You know, if you're like, there are a couple of people, hey, we really want to go over this concept, right? Would you help us with it? Sure, of course. We'll find an empty room and we'll do it. Just use the Zoom, use this link. This link will take you to my calendar and you'll see all my openings. So you pick any day and you see all my openings, you pick whichever one works for you and you put in your information. Now, let's say you want you and 3 other people from the class, add those people here, right? If you have a couple of people and y'all tried to figure out a problem and you couldn't figure it out on your own, so you asked your friends for help, which is good, and they still don't know, put everybody together. It's okay, right? We'll find a classroom, not a big deal. And then tell me about it. This is the most important thing. Share something that will help me prepare for our meeting, right? That way we make the most out of our time. All right. Let me not make an appointment for myself on my calendar. This is my office phone number. You're welcome to call my office number. This is my email. I try to be really, really quick about emails. All right, so here's our Moodle site. We have a couple of different things. This is all of our starting material, our course material. Here are a bunch of textbook links. Then I've organized the content by exams. So we have 4 major exams. We'll have 1 more chapter, and then we'll have a final cumulative exam. It is cumulative. Keep that in mind. It is cumulative. Okay. I a lot of times use the announcements forum to send out notifications to everybody. So make sure that your preferences are set where you're getting emails if something posts in the forums. So we had a few textbook questions over the weekend. So I hope everybody kind of saw that and started getting emails. Yes. If you did not, your preferences need to be changed. So you come over here to your name and you go to preferences, and then you can change it here, notification preferences. Okay. All right. Ooh, too many things on one screen. All right. I sent y'all a little welcome letter in case you misplaced it. It's right there. Everybody got the welcome letter? Yes. Okay, good. The syllabus, I'm not going to go through the syllabus point by point. I figure by this time, y'all know how to read a syllabus, right? Okay, good. So I do want to just quickly go over what is going to be graded in here. So I'm going to scroll through all of this stuff. Here's our grading policy. We have 4 regular exams, right? That are going to be worth 75% of your overall grade. Then we have 1 comprehensive exam, right? Then you're going to have homework. The homework is through the online system. As of this morning, you should be able to access all of the content material if you did the Kernel book bundle. I don't know what it's all called because this is the first semester. But y'all do know that if you join the book bundle stuff, it's only a rental. You have to give back the material, right? Okay, I don't know if people really explain that to y'all. They charge you. It's only $25 per credit hour, but it's only a rental. Okay. So the homework is through there, and then you also have reading quizzes. If I have reading quizzes listed on here, is it important that you read the textbook?
Yes.
Yes. Is the textbook easy to read? No, it's not. It's dense. It's really, really dense. Like I see a few people nodding, even in the first chapter, it's really pretty dense. So don't sit down and think, I'm going to read a chapter tonight. You're going to get burnt out. You are going to get burned out. I'm trying to be honest with you because I want you to be successful from the beginning. I don't want you to get behind. So take it one section at a time. Take a break, go do something different, right? It's dense. It really is dense, but try to read it before you come to class for that lecture. So how do you know what I'm going to lecture on? Well, let's see. Here's our class calendar. So our class calendar tells you exactly what we're going to cover in class every day. So today we're going to start chapter 1, right? So I've basically planned 3 days for chapter 1. That means that our homework is going to be due Sunday night. And then Monday, we're starting on chapter 2. Now, the homework and the reading quiz, I call it a quiz. You can have your book open. The homework, you can have your book open. I'm not, I don't want it to be, I want it to be something to where you can go and use the book as a resource. You can go use your notes as a resource, right? It's not unlimited tries. You have to try to get the right answer, but I want you to use the resources that are available to you, okay? So if you're ever wondering about when something is due, this is the calendar. This has all the dates on it. I update this daily. So if something happens, we have a hurricane, we have whatever, please knock on wood that we don't have hurricanes, but I will update it daily, okay? So don't print it out, just keep coming back to it. See if I can move this. All right. Let's see. So the other thing that I do for y'all is all of my lectures are PowerPoints, right? And so I have posted all of those PowerPoints for you. I posted them as individual slides. So these are 1 slide per page. These are the handouts where there are 4 slides per page. So whatever floats your boat, right? If you want to pull it up on your iPad, you want it on your computer, you want to print it out before you come to class, you can do that. They're all up there. Any questions on that? Good. Okay. All right. Let's see. I know you might not have this question yet, but a lot of times when it gets farther along in the semester, you're like, oh, I want to know how I'm doing. What grade do I need to make on that last exam to have a B? What, like whatever it is. This is a final exam grade calculator. It's a Sheets document that has the formulas built into it already. So the only thing is you can't just edit it. You have to download it. So a lot of times I'll get all kind of requests for access to it. Don't download it. Then you can put in your own grades and you can figure it out. All right. Our Zoom link, in case you want to join because you want the screen, even though you're in class, that's cool. There's a new COVID-19 reporting form. So the link is here. If you have had close contact with anyone who is COVID positive or you yourself test positive for COVID, this is the self-reporting link that you use to report it to the university. Please don't come to class. I love seeing your faces in class, but if you come sick, we won't be here for very long. All right? The Delta strain is much more contagious. So if you are sick, you can spread it to a greater number of people. So please, 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 if you feel sick, don't come. I'm going to Zoom. You can join on Zoom, fill out the report, go get tested. It'll be okay. Just don't come sick. All right? I promise I won't come sick. Let's do that. Yes, ma'am.
Can we also fill out this reporting form if we are sick but not with COVID symptoms? Like if we just have a cold or if we have like the flu?
I don't believe so.
But we can still use the Zoom, like that's still to our.
If you're sick, something like that, send me an email.
Okay.
Let me know what's going on. And yes, because nobody needs to come to school sick. Please don't come sick. Because you might think, oh, it's just a sinus infection, but it's not, right? Okay, so I hope that you like where you're sitting. I hope that you really like where you're sitting, because this is where you will be sitting for the rest of the semester. So it's for two reasons. One, for contact tracing. If somebody in here says, I came to class on Monday and on Tuesday I tested positive for COVID, then I need to know who is sitting around that person, right? And the other thing is, I'm going to learn your names. So I want you to go to Moodle. If you have a device, if not, you can do it when you get home, just jot it down. Here's our seating chart, right? So what I'm going to want you to do is to type your name into this Sheets document where you are. I'm going to help you out, okay? So see how this first row is row A right here. So the first seat, you just put your cursor in there. Everybody should be able to edit it and start typing in your names only in the yellow boxes. So this is A, B, C, D, E, and F. Okay, so you might have to count the number of people in front of you to know what number you are. Pretty good on that? You want me to do the letters again? A, B, C, D, E, and F. And if there's an empty seat, don't put your name in the empty seat. Make sure it's the correct number. If there's an empty seat in front of you, there should be an empty blank in front of you. I'll give everybody a minute to do that. And if you can't do it right now, just jot down your number and then do it when you have access to a device. And go to Moodle when you sit down, go to Moodle and go to the seating chart right here.
Okay.
And put in where you're sitting for me. Okay, while y'all are doing that, this is not points. You are not going to get it wrong, okay? You are not going to get it wrong. This is just for me, all right? I'm handing you back a sheet of paper. The instructions say to put your name, your IN number on the sheet, and then draw and label a water molecule. Do not look it up. Just do it, okay? Don't think too hard, just put what you know. Okay, no right or wrong. Don't talk to each other, hide your papers. Put your arm up, don't let anybody see. Don't share your answers. This is private work.
It may not be graded, but it's still private.
That's right, it might not be graded, but it's still private. I want to know what you know, not what your neighbor knows. So really hide your sheet. Don't laugh at it, but really hide it. Okay, and there might be some extras, but that's okay. I can just pass out the extras. Or if you need more, if I didn't count out enough, just wait. There's going to be some more coming at the end. I might not have counted. Okay, there should be some more coming at the end. I'm a bad counter. And those of you who are on Zoom, it's okay. Don't worry about it. It's okay. So if you haven't done your seating chart name, you should do that. And then just take a minute. And then when you're done, flip it over so nobody's looking. Hide your paper. Turn it over. Okay. No right or wrong answers. It's okay. I didn't know y'all were going to get tested on the first day, right? Everybody got a pen? Anybody need a pen? Reading. No reading. No reading. No reading. See that? See that red? Okay, I'll give y'all about another minute. Okay, anybody need more time? Raise your hand. Okay. All right, well, everybody's finished it up. The one last thing I want to show y'all on Moodle is sometimes I will ask y'all to get into groups. So I have named these groups by the elements, which is fun, right? Because it's chemistry. So not today, but in the future, you will be doing some group things, but like on your computer in a group, not get up and move in a group, because we really can't do that this semester. But so if you bring a device, that would be great. Just something that you can access Moodle with. Even your phone would work. It's easier with a computer or an iPad, but a phone will work. So if you're ever wondering where you are, the first ones are alphabetical by your name. And then these are, I want to know who else is in the hydrogen group. You can go and look and see everybody who's in the hydrogen group. Okay. So in the future, we'll be using those groups. All right, everybody ready? Pass them up? Okay, pass them up. And then if there are blank ones in the back, go ahead and pass the blank ones up as well, but just kind of separately. Everybody remembered your N numbers? I think that was probably the hardest part, right? The N number. All right, and then once you get the whole stacks, pass them this way. Pass them this way, front people. All right, Sam, pass them this way.
Sorry, I wanted to make sure we had them all.
All right. I got homework tonight. All right, and I am not going over the answer today. I'm honestly not. I'm not telling you what the right answers are today, and it's not graded. So thank you for participating. All right, so we're actually going to start talking about biochemistry. Yay, right? Okay, so if you want to follow along, you are absolutely welcome to follow along. Full screen. This way. No? Okay. So biochemistry is really kind of a new science, right? When I say new science, basically the end of the 19th century is when biochemistry really started and really got its wonderful start. And biochemistry really started with alcohol and bread, right? The great combo. So the big discovery was that you could take these yeast, right? And you could make beer and wine and bread. We knew that for a really long time, a really long time. But the surprising thing was that when you took these yeast and you broke them open, right? So it was no longer a living organism, that you could take the components that were inside of that cell that is no longer alive, and you could use it to do the same things. You could use it to ferment sugars and to make wine. So that was the magical aha moment that we don't need a living organism to do it. The little bits and pieces inside the cell will make it work. And so that's what biochemistry really is. How do all those bits and pieces work, right? That are inside of the cell. So that was a really, really, really big deal, even though I kind of simplified it. So in the first chapter, we'll talk about what is biochemistry, and then we'll talk about 3 of these really big core ideas inside of biochemistry. And these are principles that provide a framework for understanding things at the molecular level, which is what I absolutely love. So I hope that you love it too by the end of the semester. Can everybody hear me in the back? Yes? Woohoo! All right, awesome. If I ever get low, y'all tell me in the back. I try to use my good teacher voice. All right, so what is biochemistry? We want to be able to explain how something works biologically, but on the really small scale of molecular and cellular. What's smaller, molecular or cellular? Molecular, cellular. Molecular is smaller than cellular. Woo. All right, so now we define it as a core science, a core discipline. It wasn't before, but it is now. So I love biochemistry because I absolutely adore biology. It's awesome. But in biology, we learn how something works, right? Why does it happen? That's biochemistry. So then if you want to really explain biochemistry, then what science do you have to go to? Chemistry, straight chemistry, right? So you have to have a lot of chemistry. Now, if we want to explain how chemistry works, what's the next subject we're going to? Physics. Then you go to physics. So you have to go see Dr. Young for that, not me. Neither one of the Youngs, but not me. So really, what we want to focus on in biochemistry is how we can quantitatively analyze information from cell extracts, from pieces and parts, right? So we often study things in vitro, right? What do we mean by outside of a living cell? What do we usually, if I'm going to do an experiment that's in vitro, in a lab, I'm going to do it in a test tube, right? With a couple of proteins, not the whole cell. How many proteins are inside of one cell? Oh, a lot. So how sure can you be that protein A induces X result if you have 200,000 different things going on in the cell? You can't be, right? But what if I took just a few proteins out and then did that same experiment? Now I can say, yes, X causes it, or oh no, it wasn't X, it was Y, right? So that's in vitro. If we do it inside of a cell, which is also important, don't get me wrong, that would be in vivo. So in vivo is a living cell. So a lot of times the science that we do is a combination of in vitro and in vivo. So when we look at biochemistry, a lot of times what we're looking at is a fundamental relationship, and that is structure and function. How many times have you heard that before? Right? Structure and function. So structure and function, right? The way something is shaped drives what it can do. Well, what you need to happen in the cell, the function, is dependent upon the structure, right? It's vice versa. They're both dependent upon one another. And so we're going to see that all the time in the biomolecules that we're going to talk about today, especially. So, you know, using biochemistry, we can design pharmaceutical drugs, right? Because what is that going to be dependent upon? We want it to do a particular function, right? We want this to treat disease X. Well, how do we treat disease X? Well, we need to know what structure is going to inhibit a certain pathway in that process, right? So it's all about structure-function relationships. You will see that, my gosh, the whole semester. If you take Biochem 2, please take Biochem 2. It's really, really interesting. You'll see it in Biochem 2. You'll see it all over. So like we said earlier, it all started with fermentation, right? It all started with our drive to make alcohol. Find that hilarious. So normally, right, we take rotting fruit or grain. That was what was happening in the past, like 2000 BC, somewhere around there, to make alcohol using yeast, right? So that's our living organism that we were using to undergo fermentation. Well, basically, the Greeks found out that agent that was causing the fermentation was yeast. And do you know what they call yeast in Greek? Zyme. Zyme. Oh, what does that sound like? Enzyme. Ooh, right? It kind of makes sense, right? They used it to make wine, to make beer, to make bread, all that kind of stuff. So, oh, look, I was close. 2000 BC, right? All right. So we knew that this was happening for a long time. That wasn't our aha moment. Our aha moment was that we could make this reaction happen in vitro, right? And remember we said in vitro is inside of a test tube. So there was one particular guy, Buchner, who demonstrated that you could make carbon dioxide, and what is this? Ethanol or alcohol, right? Right. If we gave sugar, right, to brewer's yeast, we gave it that metabolic precursor. So what inside of the yeast were allowing that fermentation process to happen? Well, he said it must be something inside of the yeast, right? So we called them enzymes because zyme was the yeast. So it must be something inside the enzyme, inside the yeast. So it's enzymes, which I always find that's a fun thing to learn, why things were named a particular way. And there's usually some logic to it. So what are enzymes, right? How many times have you heard this? They're going to increase the rate of a biochemical reaction. If you had no enzymes in your body, you could not survive. The reactions that occur in your body are so rapid. And they are so rapid because you have a ton of enzymes doing many different things at once. So if you didn't have enzymes, none of us would be around. None of us. So in your book, there are 2 enzymes. I'm going to go back for a second. There are 2 enzymes that your book covers that are used in the production of ethanol, right? So the first one's pyruvate decarboxylase. So it will take pyruvate. If it's a decarboxylase, what does it do? It gets rid of a carbon group. So what came out of this reaction? Carbon dioxide. We're left with acetaldehyde. Now we have alcohol dehydrogenase. What's going to happen for that kind of an enzyme, dehydrogenase? Now we're talking about oxidation reduction reactions, right? Y'all are gonna love those. They're awesome. And what did we make? Ethanol. So what were our 2 products? Carbon dioxide and ethanol, right? Okay, so it's starting to make sense. So we know that these enzymes, these catalysts, are found in all living cells. All living cells. They are responsible for all of the biochemical reactions that must occur in every single living organism. So we are going to cover not all of these. Nitrogen metabolism we'll cover in Biochem 2. We'll cover some energy conversion, but not a whole lot in this one, but we'll talk about also programmed cell death. All of these are super interesting. And we'll focus a lot on aerobic respiration because it's very important to us. But you got to think, right? Catalysts, enzymes aren't only proteins. We also know that RNA can function as a catalyst as well, right? Y'all learned that in another class, right? Maybe? Yes? Not specifically? Sort of? Okay, so we'll talk about RNA as well. So how important is biochemistry in our everyday lives? Like, what does it do for us, right? There are so many applications to biochemistry, right? Basically, biochemists like to exploit the pathways and processes that happen in living cells on a daily basis. We're not super smart. We're not. We use what's already there and manipulate it, right? How do you think we design antibiotics? Do you think we came up with them? No, it wasn't us. What made antibiotics? Fungi. So when they discovered antibiotics, basically somebody tipped over a petri dish, right? And in the petri dish, he had put bacteria on the petri dish, and because the lid got tipped off, right, there was fungi that was able to grow inside of the plate. Have y'all heard this story before? Yes, right? So what happened? There was a zone of inhibition around the fungi where the bacteria wouldn't grow. So the question is, what was in that zone of inhibition, right? What was that fungi producing? It was producing, guess what? Antibiotics. So we didn't invent antibiotics. We make derivatives of what is found in nature. We modify it, but we haven't come up with our own antibiotic. That's why antibiotic resistance is such a huge deal, right? Because we can't just come up with a new antibiotic. We have to basically discover it in nature. The new CRISPR system that's out there, did we invent that? No, we found that in bacteria, right? We are not so smart that we are designing these crazy, insane things. We are just looking in nature and replicating it and modifying it and then using it to our benefit, right? So maybe that means that in agriculture, we are making, y'all know about Roundup Ready plants? The Monsanto plant around? Y'all heard about this? How do we grow crops, right? And nobody has to go out and weed these massive fields of sugarcane. Why don't you ever see anybody weeding the sugarcane fields? That's what you have to do in your backyard garden, right? Weeds will take over your garden. They will choke out your whatever you're growing. So why don't you see that? They spray something that will kill the weeds, right? Roundup. Well, why doesn't it kill the sugarcane or the soybeans or whatever it is we're spraying? Because they were genetically modified to have a resistance to that herbicide, right? So now we don't have to go out and weed the garden. Now we're going to make a ton more of the produce, right? Whatever it is that we're making, and we can feed lots of people now. So are there caveats? Are there drawbacks to these systems? Absolutely. And we have to be aware of them. But you have to weigh the pros and the cons and figure out what's the right thing to do. We're feeding way more people. Yes, we got some side effects, but we're helping a lot of people. All right, so we talked about drug design, right? You can also use it for clinical diagnostics on figuring out what's wrong with someone, right? If you understand the biochemistry of why they're sick, then you can start to diagnose them because you can look for particular markers of that disease, right? So there are so many different applications of biochemistry. I could talk about that for days, right? So in order to understand all that, we're going to have to start small. We need to start with what is the basis of things that are living? Well, that's chemistry, right? So what are the most common elements that are found in living things? You should probably be able to name the top 6 off the top of your head. There's a little mnemonic: carbon, nitrogen, oxygen, hydrogen. Okay, so it looks almost like chomps, except that M is really an N, right? So the top 6 are carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur. Isn't that kind of cool? Those are your top ones that are inside of all living organisms, right? So if you look at a human, this is their percent by dry weight, right? Because what are we really? Mostly water. So that would be oxygen and hydrogen. So if you take out the oxygen and hydrogen that's in water and you look at our dry weight, these are the elements that are the most prevalent, right? And you have some that are trace elements, but even though they're trace elements, they're super important. And we're going to talk about that. Okay, so when we look at how biochemistry is organized, right? You've probably seen this many, many times, right? If you'd start at the smallest, what we just talked about, elements, right? If you put a couple of elements together and they behave similarly, what do we call those? If you're in chem lab and you're looking at the structure of something, in order to predict what this molecule is going to do, what do we look for? Functional groups, right? What are some examples of functional groups? Methyl group. What's a methyl group? CH3, right? What else? What's another functional group? An alcohol or a hydroxyl. Same thing, right? What else? Carboxylic acid. Ooh, that's a really good one for proteins, right? Right? Carboxylic acid depends on if your hydrogen's attached or not, right? These things are starting to remember some. What else? Aldehydes, amino groups, benzenes, right? Those are some good ones. So we can put these different functional groups on our molecules and we can make biomolecules. So our biomolecules, you have 4 major biomolecules, and we're going to talk about each one of these: amino acids, nucleotides, simple sugars, and fatty acids. So these are like the building blocks of our molecules that function in the body. The ones that function are basically the macromolecules, right? So proteins, DNA, RNA, carbohydrates, lipids. We take biomolecules, we put them together, we make macromolecules, right? Because macro kind of actually means larger than micro, right? So you're going up. Then all of these macromolecules are going to function inside of metabolism. So our metabolism is all of this, the enzymatic processes that happen in the body, right? So the big ones we're going to talk about this semester: glycolysis, citrate cycle, beta oxidation is going to be in Biochem 2, urea cycle is going to be in Biochem 2. But it's all of this, right? Take A to B to C, then C can go to D or it can go to E, right? All these different pathways inside of the body that are super insanely interesting. Then when you have all those processes going together, right? Now you're going to be able to run your entire cell. So your cell is going to have all the components, right? So plasma membrane. What's our major component of plasma membrane? What? Phospholipids, lipids, all kinds of different lipids, right? You'll have all kinds of organelles that are built by the macromolecules and are products of metabolism. So then your cells, if you're multicellular, turn into a collection of tissues, which then go into organs, which then go into an organism, right? And then many organisms make up our ecosystems, right? Okay. So it's important to have a little brief review of chemical bonding in biochemistry. Our most common bonds are going to be carbon. So how many bonds does one carbon atom want to make? Wants to make 4. You have to remember that. It wants to make 4. So carbon typically wants to bond with itself, hydrogen, oxygen, nitrogen, sulfur, or I said oxygen, yeah, with those elements. And they can do that single bond, double bond, triple bond, depending on the molecule that it's binding to. But you should just kind of, I'm not going to go back and teach all this again, but you should just kind of remember, okay, this is how these molecules are made. We need to complete octets, right? Just a good remembering of that. All right. One other thing that's super important to remember that sometimes when you make a bond, if it's a single bond, what kind of bond is that? A sigma. A sigma bond has free rotation about that bond, right? What happens when I make a second bond and I now have a double bond? It's locked, right? We don't have rotation, because if I went and rotated, my sigma bond would stay together, but my pi bond would break. So that's not gonna fly, right? So our double bonds, our triple bonds don't want to rotate, because we don't want to break those pi bonds, right? So that's going to be really, really, really important when we talk about structure of a lot of different things. And the other thing you have to kind of remember is, what if you have on that molecule something that has resonance with another bond? If those two bonds have resonance, are they going to rotate? No, they're not. They're not. So not only a single double bond, but if you have a double bond that has resonance with another bond, then both of those bonds do not want to have free rotation. Okay? So kind of remember that. All right. So remember how we said that there were some trace elements, that there wasn't very much of them in your body, but they were really important? These are our trace elements. And trace elements are really important because they tend to be cofactors for enzymes. Have you heard of cofactors before? What's a cofactor? Usually an element, right?
No, yes, but usually it's like a part of an enzyme that it can't function without.
Right, so the enzyme is complete when you have the protein, right? But then it's non-functional unless it's bound to zinc, unless it's bound to iron, right? So these are required for enzyme function. They're not part of the structure itself, but they are required for function. And y'all always like stop me when we getting close to the end of class, because I have a really bad habit of like, I love this stuff and I just keep going. So, you know, stop me. All right. So ions are another thing that's really, really important for our body, especially in cell signaling and neurology, right? So how do you propagate an electrical signal? Electricity is all about charge, right? So let's say that you had a pool that is filled with pure water and it gets struck by lightning. Is it dangerous to be in? No. If it's pure water, do you have a charge? No. But does pure water really exist in actuality? As soon as you dip your foot into it, guess what? You have salts and stuff on the outside of your skin, and now you're going to propagate electricity. So yes, really, technically, if you said yes, it would be dangerous. It's true. But I'm trying to make a point. What's going to propagate an electrical signal? Ions, right? So sodium, potassium pumps inside of neurons, right? That's what's going to propagate an electrical signal in your nervous system. So ions are super, super important. If you study physiology and you study the heart, the rhythms of the heart, right? All of that muscle contraction is dependent upon what ion? Right now? Calcium. It's calcium, right? And we'll kind of talk about that in terms of muscle contraction. But ions are super important for signaling. Whether that's signaling within a cell or signaling from one cell to another cell, doesn't matter. Super, super important. All right, so y'all told me that if you took a couple of elements and you put them together and you wanted to know about function, right? You were looking at functional groups. That's why we call them functional groups. Didn't you ever wonder that? Why do we call these functional groups? Because they predict the function of the molecules that they are attached to. Right? So tell me about a phosphoryl group. How might a phosphoryl group change the structure of a protein? If I phosphorylate a protein, what What am I adding? Has to do with these little things. What are those? Negative charges. So phosphoryl groups not only add a physical actual element, a space, right? They also add a charge. So what you have to look at is all of these except for the methyl group. So all of these can have different protonation states. So these have different protonation states. Proton. That's the other thing you're going to learn about me. I can't spell. So if you ever need me to say, what was that? I can't spell. All right, so these have different protonation states. But why do they have different protonation states? Like, is it just magic that sometimes we're going to have an amino group and sometimes we're going to have an NH2? That's not charged? No, what changes that? What would make this functional group change between those two protonation states? pH, right? Well, what is pH dependent upon? Other things in solution. So we're just going to say the local environment, right? But methyl is never going to change because carbon needs those 4 bonds. Methyl's never going to change. Only has one protonation state. And what is the R? Everybody know what the R is? It's the rest of the molecule. R is for the rest of the molecule. Okay. All right. So what are our 4 big? We have functional groups, right? And the functional groups will build together and we'll make 4 major biomolecules. Let's talk about our 4 major biomolecules. Amino acids. Typically, we say amino acids are the building blocks of proteins, right? So if you look at their primary cellular function, number 1 is protein function. But is that the only thing that they do? Absolutely not. Neurotransmitters, they are parts of neurotransmitters. That's another really big one. What you're going to see a lot of times in most of them are they're involved in some kind of metabolism, right? So if you take Biochem 2, we'll talk all about nitrogen metabolism, which is super, super interesting, especially if you want to talk about like keto diets and things like that. It's also involved in energy conversion, right? You got to think about all of these molecules, all of these biomolecules we can use as fuel, right? Typically, when we're eating a normal diet, what are we using as fuel? Carbs, a lot of carbs, some fats, some sugars, right? We don't so much think about proteins, but we do. We consume proteins, we can use them. Now, what if you go into starvation mode? Guess what you can start doing? Breaking down your own muscle to use that as food, right? It's the same thing, whether it's on a cow or on you, it's still protein and you can still use it to survive, right? So you have to not forget that it's involved in energy conversion. All right, nucleotides. Everybody recognize nucleotides, right? Nucleotides come together to form DNA, RNA, so nucleic acids, right? But also in energy conversion. What's our big energy carrying molecule of the cell? ATP. We use for signaling transduction. ATP is used in signaling transduction, but a lot of the signaling transduction molecules that have nucleotides are like cyclic AMP, right? So when it's in the cyclic form, you have one thing going on, and then when it's linearized, you have something else going on. So it's a good way to have like an on-off switch. Then we have sugars, right? Simple sugars. Why do we call them simple? So that's a monosaccharide. We use them a lot in cell structure, but a lot in cellular recognition. So what do you think COVID-19, that coronavirus binds to? Something that's sitting on the outside of the cell that has carbs and lipids attached to it. That's what the virus attaches to, right? So it's hijacking a system that's already there because the thing that's sitting on the outside of the cell is doing a particular function. The virus is just hijacking it, right? Viruses are really good at that. And also in RNA, DNA, right? We know we have a sugar in there. And the last one are fatty acids, which we already said was cell membranes. We already said that we eat it. That's energy conversion. It's also sitting on the outside of cells. So cell signaling and then storage. All right, so that's a good place to end. Sound good? So next time we'll pick up where we left off. If you have any questions, if you were unable to do iClicker and need to sign in, please come and sign in. And don't forget to do your seating chart if you didn't already. Have a great rest of your first day. See y'all on Wednesday.
Sorry, what did you say about the iClicker?
Thank you, Chloe. Have a great day. Let me end this and I'm gonna help y'all. Anybody on Zoom have any questions? Looks like they all left. Okay, so I'm gonna go ahead and end.